All right. Good evening, everyone. How's it going? Excellent. Good start. Well, thank you all so much for joining us tonight. My name is Aaron Pomerantz, and I'm the Associate Director of Global Segment Marketing here at Oxford Nanopore Technologies. So I'd like to welcome you all to this special evening event. This is celebrating a decade of DNA discoveries, 10 years of the MinION in microbiology. Tonight, we celebrate the 10th anniversary of the MinION's launch, a milestone that has revolutionized the field of microbiology through nanopore sequencing. Over the past decade, the MinION has made genomic technology more directly accessible and has empowered scientists to make groundbreaking advancements from real-time outbreak response to comprehensive genome characterization. The first of its kind handheld device has inspired scientists to bring the sequencer to the samples. And indeed, many of you have taken it to some remarkable places, both on and off this world. In the lab, in the classrooms, and even aboard the International Space Station. So we see a few examples here, usage in the field. On the right-hand side, we see a student at UC Berkeley. She's just about to load her first flow cell. Perfect job. Excellent execution. They have no fear, so no bubbles. You see some amazing examples here. We've got researchers who have taken this out into the Arctic. We've got the Ecuadorian Amazon. That's a shameless plug. That's a snake that I sequenced out in Ecuador. Bothriechis schlegelii is the species name. And of course, we've got Kate Rubins, who was the first person to sequence DNA up in space aboard the International Space Station. We're privileged to have distinguished speakers with us tonight who have been at the forefront of many such innovations and use cases. We'll hear about their experiences and perspectives over the years implementing nanopore sequencing for microbiology and infectious disease applications. Following their presentations, we'll engage in a panel discussion and a Q&A from yourselves in the audience, where our experts will reflect on the field of microbial genomics and the evolution of sequencing methods and strategies over the past decade. Now, you may have also noticed over here, we've got some sequencing instruments that we took with us. So, actually, Carly and Anna, can you please raise your hands? So, they have prepared a live sequencing demo for us tonight. Which is going to be fun because we're going to ask you to guess what the mystery microbe is that they're actively sequencing right now. So throughout the talks, keep an eye out. We'll be dropping hints as to what the species is. And the winner, the first person who guesses this mystery microbe correctly, will win their very own free Mark 1D tonight, which will be cool. This is the next evolution of our Mark 1B instrument, 10 years on. So please keep an eye out for QR codes on some of the upcoming slides. You'll have 3 opportunities to guess what this microbe is. Now, without further ado, let's begin our journey through a decade of DNA discoveries with nanopore sequencing. Please join me one moment in welcoming Dr. Josh Quick. Josh is a UKRI Future Leaders Fellow at the University of Birmingham, developing novel methods for rapid antimicrobial resistance prediction. He traveled to Guinea in West Africa and established the first mobile laboratory to perform viral surveillance during the West Africa Ebola epidemic. He also developed an amplicon-based sequencing method, which has been widely used for Zika, yellow fever, dengue, chikungunya, Ebola, and SARS-CoV-2. He is currently focusing on bacterial single-cell technology as an approach to tackle the growing threat of resistance by rapidly identifying resistance genes in clinical samples. His talk title, A Decade of Nanopore Sequencing for Real-Time Outbreak Response. Welcome up, Josh.
Hi everyone, it's really great to be here. Thanks so much for inviting me. It's been quite a nostalgic, looking back through all the slides to put this together. It's been really kind of enjoyable, and I can't actually believe it's been 10 years. But the this is our first, the first time when we joined the MAP program. This was the first time we plugged it in, ran the test script, and that was April 2014. And then fast forward 10 years. This is a photo taken a couple of weeks ago by my PhD student Chris Kent in Entebbe, Uganda, with the MinION Mark 1D. It's incredible that it's been 10 years between these two events. But I was looking back through all the old slides, and our actual first publication, if you like, or Figshare article that Nick posted up was on the 11th of June 2014. So it was 10 years ago on Tuesday since we did this first ever sequencing run. And the first run that we actually did, the first real run, was a Pseudomonas DNA sample from an outbreak of hospital acquired infection that we've been investigating. And this was the only 2D read that Nick could find to map. And it actually turned out to be like quite an interesting read, that covered the O serotype antigen region. But yeah, and this is 10 years ago. And I've put in this slide here, there's been, an incredible rate of development over the last 10 years. These are the three MinIONs on the left for the geeks. That was the MAP MinION, then the Mark 1B, and then the Mark 1D, and that's the first 6 versions of the Nanopore ligation kit. And if this was like football stickers or Pokemon cards, that one on the top would be incredibly rare, because that was an unreleased kit, actually. So I was just looking back, and the actual rate of development over the first year or two was absolutely incredible. So the first flow cells shipped in June 2014. And that was the R6 flow cells, followed by R7 in July, R7-3. Then there was, by the time we got to February 2015, the accuracy had already increased substantially. We're talking about the R7-3 chemistry with the sort of 2D protocol was already giving sort of 90% identity reads even back in March 2015, which is actually incredible if you think about it. And then there was a really rapid, development through into R9. And one of the things that, we were really lucky to be a part of the MAP, and it was, a really welcoming, really great community of people that all wanted, all had similar ambitions and wanted to do, really interesting things with the MinION. But because the technology was changing so quickly, we found that we could actually, impart knowledge and try and teach people how to use the MinION. So we started this course called Pore Camp, and there's sort of been a rolling cast of people that have taught it, but it always was, a core group of Nick, myself, Matt Luce, John Tyson, and various other people like Mick Watson, Claire Lonsdale, Lauren Cowley, and everyone did. And we've done it in some amazing places. We got to travel, to some incredible places. We did it in Birmingham, which is obviously amazing. Falmouth, Singapore, Vancouver. Where else did we do it? I think, yeah, Melbourne. Yeah, so we were, as a young PhD student, it was just an unbelievable opportunity to be able to do these, travel the world, teaching people, doing the library prep basically was my job, teaching people how to do things. And people used to bring their own samples. So, and actually Anna was on one of the first Pore Camps. The next speaker, she was on the Falmouth Pore Camp, and do you remember we sequenced a crab that we found on the beach? Yeah, and there was quite a lot of other things that happened. We sequenced a scorpion in Texas, and lots of other shenanigans. But yeah, it was just an incredible opportunity to be involved with as a young PhD student working with nanopore sequencing. So I'm going to. Pardon? Yeah, so this is actually at the Salt Lick Barbecue in Texas. Jester King. Jester King Brewery. Sorry, we went to the Salt Lick Barbecue as well, but yeah, this is the Jester King Brewery, and that was like a spontaneous fermented beer, and we were sequencing it there. Yeah, exactly. Well spotted. So so rewind back to so early 2015. I think this was April 2015. We'd been wanting to use the MinION to sequence samples, Ebola samples in West Africa. We wanted, obviously it was quite difficult to transport samples out of West Africa, so we wanted to use to put together a mobile lab with the MinION and go to West Africa and do the sequencing there. So that's what I did. That's the, you might have seen this picture before. That's basically all the, the portable lab all packed up at Heathrow. And then on the right, that's the first sequencing run. And if you're a real OG, if you remember that Minnow display there with that histogram, and that was one of the first ones. But I mean, look how amazing these flow cells were. So we were doing these are some of the first runs that we did. The old Minnow displays to have all the 512 channels, and it was, a sea of green on these runs. So these are obviously a really good batch of flow cells. But the point of what I wanted to show here is, if you look on the right, that's the PCR run. This was singleplex amplicons. We had 19 amplicons in this experiment to generate coverage, tiling amplicons, singleplex RT-PCR reactions, tiling the Ebola genome, and then one flow cell per sample. So we were basically at this time using pretty much the sort of entire supply of MinION flow cells to do this stuff to do this sequencing. So obviously and then back then there was no local base caller. So I used to sort of do the sequencing run, get about 500 megabytes of data, and then I'd go back to the hotel and try and upload it using the hotel Wi-Fi connection, so that Nick, and I don't know if anyone remember this is metricore.exe. So you had to basically, this was like all of the base calling happened in AWS. So you had to sort of use this client to upload the data, download the data. And we'd basically we'd upload 500 megabytes of files and download the reads, and then Nick would analyze the data back in Birmingham. But yeah, we managed to generate coverage. These are the, some of the early versions of the scheme covering the Ebola genome using quite long amplicons. And then look at this. This again is the sort of data that we generated from those early sequencing runs. As I said, this was the R7-3 chemistry with 2D. It's actually really good, and you can call, look, it's like over 90% identity to the genome for the PCR data, and you can easily call the SNPs from this data. So that was all the way back in April 2015, and we identified that there were these two, very early on, that there were these two lineages circulating in Guinea at that time, which wasn't known because there hadn't been any sequencing data released for a long time. And this was the total number of the sort of headline figures from our paper, which we published. And we did about 170 flow cells in total. It was at the end of the sort of epidemic, the sort of tail end of the epidemic. And we got, over time, coverage increased. And the turnaround time that we managed to achieve on the right was, basically 1 or 2 days turnaround time from sample receipt to generating the genome. And if it was longer than that, it was because we were going back and getting samples out of the freezer and things like that. I haven't got time to talk about this, but this was from this we were able to use the genome data to add information to the ongoing epidemiological investigations that were looking at the contact tracing, the chains of transmission that were circulating. And this is the sort of overall phylogenetic tree. And it was the first time that genome data had been used in a WHO situation report. So this was October 2015 when that happened. And this is a timeline from Verity, a review by Verity Hill in Nate's lab. And since the Ebola epidemic, there's been a number of outbreaks. Some of them epidemic. Some large outbreaks. And then obviously the COVID-19 pandemic, and every single outbreak since that Ebola outbreak, nanopore sequencing has played a bigger and bigger role in the outbreak response, to the point where for Ebola there was 30,000 cases, of which 5% were sequenced. So there were 1,700 genomes released for Ebola, and that was considered an amazing opportunity, an amazing achievement at the time. But during the SARS-CoV-2 pandemic, there were 15 million genome sequences generated by over 4,000 labs. It's the biggest sequencing, collective sequencing effort, in the history of mankind. So it's incredible, and obviously nanopore sequencing was a huge part of that. So anyway, just to finish off, I was going back through Nick's 2014 London calling talk called A Year of Happy Mapping, and it's on YouTube, and you should go and watch it because it's only got 400 views, and it could really use. And I found that this really random slide where he said that he'd gone to Salisbury, and on the way past he'd gone past Stonehenge, and he said generations of archaeologists had missed this detail here. So I wanted to do an experiment and see what 10 years of research into a decade of research into generative AI has done. So I put in this prompt, and I got this. So anyway, thank you very much for having me.
That was fantastic. Thank you, Josh. Now, let's see. Before we move on to our next speaker, I think we can switch over and go ahead and take a look at our live sequencing demo. We're going to start to switch over to that screen. Excellent. And I'm going to hand it over to our application scientist up in the Bay Area, Carly, to walk us through what's going on with this live sequencing run.
Looking at here is our Minnow user interface. So right now you can see an overview of the run. So it's been running for about an hour and 7 minutes, and we're looking at about 99,000 reads at the moment. On the next slide here, you can see our channel states panel. So this is actively looking at all the pores on the flow cell, and green is good. Green means that there's DNA actively going through the pore at the moment. For pore activity, you can now look at what percentage of the sequencing pores are active. So we're at 84.3% sequencing. And then here you can see a read length histogram. So right now our N50 is at about 12.8 KB. And then you can also see your reads compiling here. So this is a total throughput. So total reads here is about 95K. And then here's the trace viewer where you can actually see the DNA going through the pore and what that would actually look like. So now we're gonna you can actively start guessing. So this is hint 1 for this, is it is a gram-negative bacteria. Hint 2 is that the genome size is approximately 2.5 megabases.
Broad hints.
And back to you, Aaron.
Thank you, Carly. So we're gonna switch back. And if you haven't seen one of these before, we're going to see a QR code. So how this works, if you feel like you're ready to guess based off of those 2 hints, gram-negative, genome size about 2.5 megs, then you can scan the QR code and you can submit a guess. Now, your guess will be anonymous if you don't add your name or at least your first name and your last initial. So if you would like us to know who wins at the end, please add that in your free text field. Otherwise, we won't know. It will be anonymous. So I'll give a minute. Go ahead and scan that. And you can submit your guess. And we'll have 2 more opportunities for hints from Carly, and they'll get progressively a little closer each time. All right. With that, next up, we have Dr. Ryan Wick. Ryan is a bioinformatician specializing in long-read assembly of bacterial genomes and has been using the MinION sequencer since 2017. He has developed numerous bioinformatics tools, including Bandage, Unicycler, PoreChop, FiltLong, BadRead, Tricycler, and PolyPolish. Ryan earned his PhD from Monash University under Professor Catherine Holt and is currently a postdoctoral researcher in Professor Tim Stinier's group at the University of Melbourne. His talk title is Perfect Bacterial Genomes from Nanopore Reads Alone. Welcome, Ryan.
Yeah, so I have been working with bacterial genome assembly for almost a decade now, and for most of that time I've been using Oxford Nanopore reads, and I've really been chasing this idea of a perfect bacterial genome assembly. And I know that's a strong word, and some people don't like that word, and a question I often get is there really such a thing? Can you really get a perfect assembly? And I would make the case that, yes, it is a thing, and in most cases, it does make sense to talk about a perfect bacterial genome. If you're producing an isolate culture that came from one cell, well, that cell had a genome, and as it divided, yes, there might be some mutations, but they will probably be in the minority of the cells. And yes, there might be some dynamic regions of the genome. And so maybe we just need to be a bit more careful about defining what I mean by a perfect genome assembly. Well, I think a perfect assembly should obviously be complete. It can't be fragmented in any way. I want no errors at all, no substitutions, no indels, no rearrangements, absolutely nothing. Really what I'm going for is the sequence of that first cell that started your culture. But yes, there can be dynamic regions of the genome, and that can add some heterogeneity to certain regions. And so I will say that whenever you encounter those, you go with the majority. For a simple example, if there was a locus in your genome where 70% of the DNA says C and 30% says A, well then C is the correct answer that should go into your perfect assembly. And I think for most bacterial isolates, this holds up, and a perfect assembly does exist. I started working with Oxford Nanopore in 2016. I started at a Poor Camp, where I think that's where I first met you, Josh, I believe. And I dug up some of the reads that I made back in 2016. This is from a Klebsiella isolate. And the reads were pretty rough. I think this was probably just after the switch to R9, but they were 1D reads. Yeah, a bit unwieldy, 1 read per file, and as Josh said, you had a base call with Metricore on the cloud. Read identity was pretty shaky, more than 10% error rate on these reads. And then an assembly that you can build just using these Oxford Nanopore reads was also pretty rough. Almost 2% error rate. For this Klebsiella genome, that equates to about 90,000 errors in this genome. And that means that every single gene in this genome had lots of errors. It makes things like annotation extremely difficult to impossible. And before I move on, I want you to note that quality score of about Q18 for the assembly quality that you could get back in 2016. Hold that in your head for a bit. These very low quality reads also created a huge bioinformatics challenge because a lot of the algorithms that were developed used things like k-mers that assumed you could get pretty long stretches of accurate sequence, and they completely didn't work on reads with more than a 10% error rate. So this resulted in a huge burst of algorithmic development. And there were all these papers coming out using new algorithms that could work with such error rates. And can you see what everyone's favorite word was in this era? Noisy long reads. Noisy, 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 noisy. So many new algorithms came out for these noisy reads. So how would I generate a bacterial genome assembly back in 2016 using such noisy reads? Well, I couldn't rely on Oxford Nanopore reads alone because, as you saw, that just creates too many errors in the assembly. So I developed Unicycler, and Unicycler leans on Illumina reads first to build an assembly graph. And then it uses the Nanopore reads, the noisy reads, so I don't think I used that term in my paper, to scaffold it out. And they don't actually have to be very accurate because all you're using the reads for is to untangle the graph. And this does a pretty good job of generating a complete bacterial genome assembly, but it's become increasingly clear to me over the years that it is not going to give you a perfect bacterial assembly. There are some errors that tend to creep their way into a unicycler assembly. So let's fast forward a bit. In 2021, which at least for me is kind of the mid-ONT era, I've got another Klebsiella isolate to use as comparison. Things have moved on a bit. Thankfully, we don't have 1 read per file anymore, and we can base call locally with Guppy. And the reads have gotten considerably better, much less than 10% error on most reads now, about 5% error on most of the reads. And the assembly that you can generate from just those Oxford Nanopore reads has gotten a lot better too. But still, there are a lot of errors in there. Almost 2,000 errors in the entire genome, which means that more than half of the genes in this Klebsiella genome are now perfect and error-free. So we've come a long way, but still a lot of issues. You can see homopolymers are still creating issues, and they create frame shifts, and that does make annotation difficult. So how would I assemble the best possible bacterial genome in 2021? Well, since Nanopore assemblies had gotten so much better, I leaned away from Unicycler's short read first approach to assembly to using long reads first. And this was when I developed Tricycler, which can give a very, very clean consensus assembly using nothing but your Oxford Nanopore reads. And then I would just bring in Illumina reads at the end to polish it up. And I developed PolyPolish to do that. And I think in 2021, maybe this could give you a perfect genome. Possibly. I wouldn't guarantee it. Certainly a better genome than you can get out of Unicycler, though. So we're making progress here. All right. Today. This one was just from a few weeks ago, maybe about 2 weeks ago. We sequenced another Klebsiella. And the reads have a new file format, new base caller, and they have gotten so much better. Massive increases in read accuracy. These are just simplex reads, one single molecule, one pass. And recall I asked you to remember that accuracy score of the assembly was around Q18. Our reads are now about Q22, which is to say a single read, single molecule, one pass from today is considerably better than our best assembly we could have generated in 2016, which I think is incredible. Now, the assembly that we can generate from these has gotten very, very good. I made this assembly, and I think there are 3 errors in the whole genome. That is all. And only one of them is an indel. So we are nearly at the point of perfect bacterial genomes from Oxford Nanopore reads alone, and I definitely have seen cases where I get a perfect genome only from Oxford Nanopore reads. So today, how do I go about doing assembly? Well, I largely use the same approach that I did back in 2021, but the changes I will make is we've gone from like maybe you can get it perfect to I think you probably can get it perfect now. In most cases, I think you can assemble a bacterial genome to absolutely no errors. And the other big change is that Illumina polishing step used to be pretty important when you were fixing hundreds or even thousands of errors. Now you're fixing very few, so I still do it if I have the Illumina reads, because, you know, if there's 3 errors, I'd like to fix them, but it's just not as important as it used to be. And the final thing is Tricycler, while it gives very good assemblies, I don't know if anyone here has used Tricycler. It's kind of a lot of work. It's a bit unwieldy, and I don't find it to be a very elegant solution. So this is kind of what I'm working on at the moment, is we need a replacement for Tricycler that essentially does the same thing, but is much more automated and much easier to use. And so that's in progress. Stay tuned. Looking into the future, well, I think Oxford Nanopore accuracy is going to continue to increase, and that is just going to continue to make things easier for us. We are going to develop new tools and new best practices, and this is largely what I'm working on in the near future, is really trying to get us to the point where we can easily get a perfect bacterial genome assembly, at least most of the time, for most of the genomes that we work with. Looking a bit further out, getting a bit more speculative here, well, I still feel confident in predicting that accuracy will continue to go up. And I would like to get to the point where we can easily get perfect assemblies almost all the time. There's always going to be the occasional outlier, difficult bug that gives you problems, but I want most of our assemblies to come out absolutely error-free. Can we get rid of short reads altogether? That's a good question, and I don't know, but I would love to. And so I've got my fingers crossed there. And then I think maybe the final frontier for bacterial genome assembly is, remember I said at the start, yeah, there can be dynamic regions of the genome, there can be variations. Let's just go with the majority variant to keep things simple. Well, I think assemblers and tools that really characterize what's happening in those dynamic regions, and don't just throw out the minority variants, but include them and describe them, that's going to be very useful. It's going to be incredibly useful for metagenomics, where this variation is so common, and that I think will be the next big frontier that will give us a lot to work on. Final thing I'm going to say is much of what I've said here in the future, maybe most of what I've said here, the onus is really on the software developers and the tools. The reads are good enough. The data is good enough. I think we've gotten to a point where it's the bioinformatics that needs to catch up now to really make use of the data that's now available. So, thank you.
Thank you so much, Ryan. That was fantastic to see the evolution of sequencing accuracy over time in bacterial genomes. And I think one interesting thing to call out, too, is that some of the recent gains have not been with the chemistry themselves, but really just the machine learning teams really just reading out that signal and making those improvements just on the data that's been generated. So with that, let's go ahead and switch back over to our live sequencing demo, see how that's doing. And remember, in a moment, we're going to show that QR code. So be ready after Carly gives us a couple more hints. Over to you, Carly.
So we're at 122,000 reads now. still sequencing, and our N50, which is our median read length, is about 12.8 kb. But I would like to point out that we are getting really long reads, so we have a long read of about 80 kb. So our next hint, hint 3, it is a plant pathogen. Hint 4, it is transmitted by insects. Back to you.
All right. Thanks, Carly. Let's go ahead and flip to that QR code. Feel free to scan that, give a guess. A plant pathogen, 2.5 meg genome, transmitted by insects. We've got a lot of microbiologists here, so I'm guessing someone's going to get it. Out of curiosity, does anybody feel pretty confident right now, if you've done a guess? Not yet? Yeah.
And please include your name in your submission.
Yes, thank you for that reminder, Carly. All right, we're going to get one more shot at that with a couple more hints in a moment. Up next, we have Dr. Anna Cusco. Anna is a postdoctoral researcher in the Big Data Biology Lab at Fudan University. She is currently characterizing Shanghai pet dog microbiomes using long-read metagenomics. Anna earned an industrial PhD at the Universidad Autónoma de Barcelona in 2017 in collaboration with the Vetgenomics company. During her PhD and afterward at Vetgenomics, she explored animal microbiomes using multiple long-read sequencing approaches. Her research focuses on the canine microbiome and the application of novel technologies to characterize it. Her talk title, From Fur to Feces, Exploring Canine Microbiomes and Beyond Using Nanopore Sequencing Strategies. Welcome up, Anna.
So thank you. First, nothing of this has influenced my results. So let's talk about why should we care about dog microbiome? Because we care about our pet health, right? So we know microbiome has a lot of influence in dog health. Because One Health, we do not only share spaces with our animals, we are also sharing probably some of our microbes. And then we have also seen that dogs seem to be very good models, especially for microbiome, for gut microbiome and skin microbiome. Pet dogs seem to be more similar to humans rather than other animals that are commonly used. So how we have been analyzing, or how can we analyze microbiome? We can analyze it using amplicons. This means that we are using specific marker genes. For example, in bacteria, 16S is the most common one. So these marker genes allows us to classify our community in terms of taxonomy, but we cannot see anything else rather than the kingdom that we are targeting. On the other hand, we can also do a metagenomics approach where we sequence all the material, all the DNA there. And if we got enough depth, we can get the metagenome assembly genomes. These are genomes, so we can get information about the antimicrobial resistance, about the potential functions of the genomes, and we can get everything out there, not only bacteria or specific things. So this is mainly a summary of my whole research career. It has been dogs, nanopore, and microbiome, I could say. So yeah, I started with the skin microbiome, which is a low biomass sample, and it has a lot of complexity, so there is a lot of stuff there. And now I'm mostly in fecal microbiome, which is high biomass, so it's nicer to work with, especially for the microbiome, and it's medium complexity. This was my first London calling, not my first London calling, I think my second one. So yeah, first we started for the skin microbiome. As I told you before, it's a low biomass sample, so we had to go for an amplicon approach. I have done before the short-read amplicons, but short-read amplicons can only give you information at the genus or family level. And then we wanted to try long-read amplicons using Nanopore. So we targeted the full-length 16S and the 16S ITS 23S in order to try to reach deeper taxonomic resolution, in order to try to get genus or species taxonomic information. We did that in a couple of samples, in a chin sample, in a dorsal sample. That's what we found. There was a lot of Pseudomonas in the chin, and then we detected specific species. And we finally saw that we're probably just sequencing the water that the animal has just drank, because it was very environmental stuff. So yeah, again, we are confirming like dog skin microbiome is really low biomass. So we somehow decided to switch strategies. So we went for culture things. We focus on Staphylococcus pseudointermedius because it's a common commensal on dog skin, but it also is the main causal agent of canine pyoderma, that it's a skin infection of dogs. And some concerns about this is that it can have methicillin resistance and it can have zoonotic potential, so it can be transferred to humans. We detected, what we did is assemble whole genome sequencing with Nanopore only, and we got the genomes and the plasmids and everything. And what we saw is like we detected this Staphylococcus pseudointermedius that has methicillin resistance in healthy animals. Generally speaking, the genomes were larger and they harbored more antimicrobial resistant genes. And the next step that we are working on in collaboration with, still with the Barcelona team, it would be to do this rapid clinical metagenomics assay for the canine skin infections, which is like the great thing that has Nanopore, right? So the idea is working together with the veterinarian clinicians. They will diagnose the pyoderma in the animal, then they will sample, and we will process the sample, and we will get a rapid response, a report with some information regarding the species, the antimicrobial resistance genes, and maybe we can assess them faster to say which antimicrobials could work for that specific infection. So now let's move from food to feces. So now let's move to dog gut microbiome. So what's out there? I mean, this is mainly my postdoc project. So most of the studies out there are based on 16S rather than metagenomics. In fact, when you look at metagenomics studies on dogs, there are just 5 studies that have more than 10 animals characterized. All of them use short-read sequencing, and only few breeds are characterized. And they are usually not pet dogs. They are dogs from nutritional companies or maybe training centers. So yeah, there is a lack in characterization of dog gut microbiome of pet animals. And also, last but not least, dogs are the most phenotypically diverse mammals. Both Chihuahua and Great Danes are the same the same species, right? So yeah, I wanted to do that, of course, using long-read metagenomics. So the first thing is getting these long reads. Then the idea is to get the metagenome assembled genomes. The good thing about using long reads is that you can get these super nice single contig max, which is great. But of course, not all of them are single contig max, which that is the ideal thing that you can have. But of course, in the metagenome, you also have like very long contigs that you still need to assemble further. For that, you can do that experimentally using some kind of high-C proximity ligation approach. We did that before with one sample, and now we are doing bioinformatics binning using semi-bin2, which is developed by Xiaojun in our lab, to kind of bin all these long contigs into max. So that's what we have right now. So I have a total of 52 pet dogs in Shanghai. I have been around Shanghai collecting samples from dogs. These are some of the volunteers. We have also a lot of extensive metadata, like more than 80 questions, like 20-minute questionnaire that the dog owners had to fill in. And we got 20 gigabases of Oxford Nanopore data and 20 gigabases of short-read sequencing data. And then we did, the bioinformatics binning. So that's what we have right now. So we were happy to see that. So each one of these bars represents a sample. So we can say that we got more high quality max than medium quality max per sample, which is great. And also some of them were even single contig. So the median was like 36 high quality and 12 medium quality max per sample. These are the total number of max. When they're replicating at the species level, we got 320 species level max. 80% of these were high quality and 20% were medium quality. And from the high quality, like half of them were single contig and the other half were several contigs, but still 4 contigs per max. So very, very contiguous assemblies. And another thing that we check is like we mapped back the raw reads to our species level collection, and we saw that almost 90% of the raw reads were mapping back to the max, which is great. I mean, we are capturing all the metagenome in max. We are also applying that, I mean, this is the project for Ichian one here in the picture. We are applying that also to soil samples in urban parks with a little bit more of the data, so 40 gigabases of data instead of 20. Soil is much more complex, so results are not so nice as in dogs, that it's like medium complexity, but still we are getting a lot of high quality max and a lot of novelty things. So yeah, this is it. So some of the conclusions is like with Oxford Nanopore, especially when we were in vet genomics, it was very good because we could profile in-house the microbiome and the microbial isolates, and we didn't need an external service provider, and we could play around with different approaches and test a lot of different stuff, which was very cool. Then long-read amplicons allowed us to profile at the genus and the species level. We also detected these Staphylococcus intermediates that are methicillin resistant in healthy animals. And finally, with the long-read metagenomics, in our case in dogs, we were able to retrieve this highly contiguous max, even single context, and capturing the whole diversity in max. And yeah, that's it. I would like to thank both my colleagues in the postdoc in China and then the Barcelona team. These are our t-shirts that we did for London Calling. I don't know, remember the year, but we did like a specific MinION t-shirts for going to the London Calling. And yeah, I would also like to, of course, thank all the participants. And yeah, thank you all.
Thank you, Anna, for that wonderful talk. Now, moving on, we've got our last couple hints coming up for our live demo. So I'm going to pass it over to Carly for our last two hints. Take it away.
We're at 141,000 reads now. Hint 5. It damages the water transport system within plants, including a prevalent crop in Georgia. Hint 6. It can cause visual clues of infection, including leaf scorching. Back to you, Aaron.
Thank you, Carly. All right, let's pop the QR code back. Go ahead, take a snap. Let's leave that up. Give folks a minute to guess. So those are our last two clues. I'm just curious, does anyone feel confident now? Maybe. We'll give the answer at the end. Excellent. Okay. Now, finally, I'd like to extend a warm welcome to Dr. Matthew Keller. Matt is a biologist with the genomics and diagnostics team within the CDC's influenza division. He joined the CDC in 2017 to develop a portable sequencing pipeline for influenza A virus outbreak detection and pandemic preparedness. During the COVID-19 pandemic, this work naturally expanded to SARS-CoV-2 surveillance. Both sequencing pipelines are currently being deployed to international partner laboratories to expand access to influenza virus and SARS-CoV-2 surveillance. Over to you, Matt.
Thank you, Aaron, for that introduction, and thank you all for joining us today. I also have a disclaimer that none of the names or commercial anything I say is an endorsement. And I decided to say this because Jim wouldn't buy me a motorcycle to add a portable sequencing platform too. So thanks for nothing, Jim. So our team its main focus is on the genetic sequencing of influenza viruses. We're coming up on 70,000 genomes, and there's a lot of data in projects in here. The main one, and really the reason why the team exists, is to monitor human seasonal influenza viruses and make recommendations to WHO for vaccine strain selection. But humans aren't the only animal to get influenza. In fact, we're not even the natural reservoir. Waterfowl are the natural reservoir. And they harbor a great deal of genetic and antigenic diversity. And they do a lot with this diversity. Influenza can jump species, it can reassort segments as it goes. And as much as I would love to go to Antarctica to swab some seals, most of the action is sort of on the right-hand side of this slide. Including lately, of course, H5 that has made its way into cows, a response in which I'm heavily involved in at the moment. So when I joined CDC, a lot of these sort of zoonotic events where an animal virus can jump into people and spread quickly was sort of picked up in those centralized high throughput sequencing strategies after it's made it to people. We were curious if we could sort of move that detection point up and look for viruses at the human-animal interface. And again, this is sort of a summary of my entire postdoc when I joined CDC was, can we take the work that goes on in this building and shrink it down in size to a suitcase or a couple of suitcases and take it into the field and do that kind of surveillance. So we did that. There's a lot of modifications to the workflow, including switching to the MinION sequencer. So we created this portable sequencing lab in a box and took it on the road. One of the places we decided to sort of test it out was at swine exhibits. Think like Westminster Abbey dog shows, but for pigs. It's a very interesting event if you get a chance. Don't go. But there's a lot of travel to and from these shows by the pigs. They go to a lot of shows. There's a whole circuit that takes place over the summer, about now. And unlike sort of agricultural pigs that are raised in isolation, there's a lot of contact between people and the animals and moving around. And there is almost always flu there. So that's where we went. We went to a very large show. There's about 3,000 pigs at this particular event. And we set up in the barn. We swabbed pigs, sequenced overnight, and analyzed the data right there. And we definitely found the flu. We found human seasonal viruses, which is what we expected, honestly, but most of our samples were a bit more interesting. This was an H1 Delta 2 virus that is descendant from the old human seasonal H1N1 virus. Now that virus got displaced out of humans by the 2009 pandemic, but it didn't go extinct. It actually jumped back into pigs and circulated there, accumulating mutations and evolving and moving further and further away from the old vaccine. So we saw this virus that hasn't been circulating in people in a while. There's a lot of kids at this show, and it was a poor match for the nearest vaccine. So we actually emailed some Nanopore data back to our colleagues at CDC who began synthesizing a prototype vaccine. And that email took place about 16 hours after we opened the cases and set up and started work. This virus actually ended up jumping into people. We detected it a few months later through our routine surveillance efforts, and that vaccine has now sort of been upgraded to an official pre-pandemic vaccine that's in the freezer at WHO somewhere. So we have this lovely influenza lab in a box. We deployed it a few times to do some animal surveillance. We actually deployed it to do human surveillance. This whole state of Louisiana was down for a cyber attack. So we took this there and did some sample processing there. But could we sort of build a companion module, if you will, to another respiratory virus, specifically SARS? And so we did that. We actually decided to focus on just the S gene for a lot of reasons, but one of them is primer conservation. If you look at the diversity and mutations of this S gene, this really important region of the SARS genome, there's a lot of diversity and mutations. It's sort of a minefield if you're trying to get primers to anneal there. This strategy did pay off. We designed our primers initially during the beta wave, and they've remained over 99% conserved at every position since. But you know, the laboratory tools are only half of the story. The work obviously doesn't end once you hit start on the sequencer. So we needed an analysis suite to go along with that. And that's where MIRA comes in. And MIRA is our flu and SARS assembly and QC pipeline. Its scope is a bit beyond just some of the tools that I'm showing here, but it is what we use to analyze the data. And the idea is to be a surveillance tool pipeline to go from, you know, that FASTQ directory to something that's ready to get uploaded to a public repository. Our lab protocols are available by that link if you're curious about them. So we have all these great tools. We've been to the barn. It's time to sort of share them a little bit and travel around. So our strategy for trainings was a regional approach. Our first one was in Thailand. And this is really a 5-day intensive hands-on workshop. And we have a collaborating center there, and we invited, for this one, 20 representatives from 11 countries to come learn how to do this. There were quite a bit of lectures. There was 10, maybe too many. There was 10 lectures that really cover everything from sample selection, how the lab works, how assembly works, and how to upload data. And importantly, how to do, you know, data curation and QC and all of those good things to get quality data uploaded. There is, of course, hands-on experience. They tested real samples, generated real data, and we copied that data onto all of the laptops that they brought and let them analyze all the data themselves. One, to get the experience running the analysis software, but two, to make sure the install worked. If you've tried deploying, you know, some of your own developed software, you know that there is a great diversity of computers out there that people use, and that is certainly a challenge in this. But once the training is done, the work doesn't end. We want these people to go home and actually do this work. So we set them up accounts with Nanopore and our international reagent resource and ship them primers that we've manufactured. We also ship them polymerases, library chemistry flow cells, even a Mark 1C sequencer, and of course provide software and technical support. This is a very active process where we are tracking the accounts and the shipments and the data of all the countries that we are supporting in this effort. Since Thailand, we haven't stopped. We've done 7 regional trainings, 136 people from 84 countries. We've basically made it all the way around. I am headed to Georgia in Eastern Europe on Saturday, next Saturday in a week, to do our 8th training. And this has been a very fruitful project. We have thousands of genomes, genetic sequences uploaded. We're building a network for surveillance and building local capacity and, you know, empowering these countries to do their own surveillance. But another sort of result from this is the training program itself. Since COVID, there's an entirely new division at CDC, and they are using our training experience to build their own training program and how to teach countries to do surveillance and, you know, expand this to even more pathogens beside just flu and SARS. So this is ongoing work. We're going to keep doing hands-on training, and we are going to train the trainers on how to put together a program like this. This was a huge project, a very long-term. There's a lot of people to thank. I want to thank all of you for listening, and finally want to plug that I am looking for a postdoc. Thank you.